like a embryonic cell. The iPS cells are 99% of the way there, but that 1% is still, if you have 1% of your genes that were not regulated properly, that would be a lot of problems. Even one gene not regulated properly causes disease, right? So you want to get as close to that 100% mark as possible. And so we compared NT somatic cell nuclear transfer derived embryonic stem cells and induced pluripotent stem cells, and found that the epigenetic marks, the differences between real embryonic stem cells created by in vitro fertilization, look closer to the nuclear transfer cells than iPS cells.
Now, how much closer?
Well, so this is, we give some numbers in the paper, but some of the numbers are something like 8-fold less epigenetic differences, things like that.
So those are eightfold less things that can go wrong.
Yeah. Well, I guess they don't all necessarily have to be having a consequence. I mean, they could be differences that have no molecular consequence, but from the RNA studies that were carried out by my colleague at the UCSD, Louise Laurent, they do have an impact on gene expression. So Louise carried out RNA-seq experiments and showed that the NT cells are more like ES cells in terms of their gene expression profile. So that likely links these epigenetic marks to actual changes in gene expression, which of course would be an important difference.
Okay. What about parthenogenetic stem cells? Did you look at those?
No, we didn't in this study. These were all derived as part of a study actually that was published earlier by Shukrat Matalapov's group in Oregon. And so these were the published cells, and they were the nice thing from an experimental point is they were all genetically matched. So you had the same nucleus that was being delivered into an enucleated egg, and the same nucleus, same cells were being where genes were added with IPS technology.
So you can have a real apples to apples comparison.
Exactly. That's why I was interested in the collaboration, because we really could address it in a very clean experimental fashion, where you don't have to worry about different genomes being a cause, because the underlying DNA sequence can actually have an effect on epigenetic patterns.
Okay. Now, what's the implications for efforts in preclinical trials with iPS cells? There's a couple being looked at just here in San Diego for Parkinson's, for example. One with iPS, another one with parthenogenetic stem cells.
Yeah, so I think, even within the set of iPS cells that we looked at, knowing where to look now, because we have a set of regions that we know are always problematic in iPS cells. Some of the iPS cells are actually a little better than others. So if you use these regions to screen through a lot of different iPS cells, you might find ones that are closer to the NT cells. And we actually can see some of those. So not all the iPS cells show this 8-fold difference. Some show less.
Okay,
so I would say that there's, it's not a total, bad news story for iPS cells. It's just that, therapeutically, these cells, they don't have any introduced genes into them, right? So you're not adding any gene, and their epigenomes look much more like ES cells. So I would say that they would be the cell type of choice if you were going to do therapy. It doesn't mean the other ones you can't find good iPS cells, but unless you look at this level of resolution, and you might not actually be even aware that there are those differences.